So FLAG-IDA uh, has a response rate of about 50%. Complete response rate of about 42%, meaning that the 42% of patients who are receiving FLAG-IDA will be able to go on to have a stem cell transplantation. However, this is where blinatumomab comes into play. Um, this has been up until combination chemotherapy has been really the only option up until very recently. But for this relapsed or refractory group of patients who are unable to achieve CR1, complete remission, one within UCAL14, we can offer blinatumomab, which is novel, certainly in respects to combination chemotherapy. So a couple of things that we should all know about blinatumomab. First of all, blinatumomab certainly isn't chemotherapy. Blinatumomab is a BiTE compound. BiTE compound stands for bispecific T cell engagers, hence why they're called BiTE compounds. They blinatumomab itself is owned by Amgen. And they are essentially highly modified antibodies, which have been engineered to force the body's immune system, and particularly the body's immune killer T cells, to come into very close proximity to leukemic cells and start an immune response against those cells and induce remissions via the body's own immune system. So this leads us on to a couple of questions. First question is, what is a killer T cell? A killer T cell is a crucial component of the body's immune system and are capable in their own right of destroying cancer cells. They're also quite important in recognition and destruction of virally infective cells. There's long been a positive correlation between the presence of killer T cells within tumors and the long-term survival of patients. And this goes back to work studies in the 1970s and has been shown to be a positive correlation for improvement in prognosis for non-Hodgkin's lymphoma, ovarian, and colorectal cancer patients. And you can kind of think of a malignancy. You can always think of it. It's very important for a malignancy, any malignancy, including acute lymphoblastic leukemia, that it must develop the ability to evade these T cell directed immune responses, because simply otherwise that cancer wouldn't survive. However, this does lead us on to a potential mechanism of cancer treatment. And to go back to our image of hematopoiesis, the natural killer T cells are here. This is an image from Cambridge University, and this is actually of an ovarian cancer cell. And this cancer cell is undergoing, should we say, interactions with the natural killer T cells. And what the natural killer T cells are doing is almost inspecting the cell surface membrane of this cancer cell. And if the natural killer T cells identifies that this cell is cancerous, or in another case maybe even virally infected, it will start small perforations into the cell surface membrane of that virally infected cell or that cancer cell, and inject it full of toxic genzymes, which are enzymes which are involved in breaking up the cellular components within that cell. This is a very highly toxic compound, and that will result in the cell's death. This, in cell biology terms, is known as the kiss of death. So there we go, lethal stuff. Um, so now we know what a natural killer T cell is. I wanted to ask myself the question and illustrate what is an antibody. An antibody is a glycoprotein. It has two main components: the light chain and the heavy chain. And the light chain is really where the sort of business end is. Light chain is made, and right at the end there is an antigen presenting site. The antigen presenting site is a unique component which will only bind to a specific antigen. It will not bind to anything else other than that specific one antigen. So, for example, if this antigen binding site or antigen presenting site has been made for one specific virus, it will be ineffective and will not recognize any other type of virus. And that's illustrated here from sort of block images of antigens. As I said before, the cell which is responsible for the vast majority of production of antibodies are the plasma cells. And what antibodies do is they are, as I said before, tailor-made to specific viruses, fungi, or bacteria. And what they do is they are produced en masse and flood a particular microbe, and that action acts as a great attractant for other components of the immune system, such as your basophils and xanophils and neutrophils, and they are the immune system, which part of the immune system which was responsible for a phagocytotic immune response against these intruders. So this was how I pictorially represented these two components, which are critical to BITE technology, as antibodies are almost like Cinderella's shoe, in that this shoe would only fit Cinderella, but the antibodies will only bind to a specific protein found on a virus, fungi, or bacteria. Now, natural killer T cells are these components of the immune system which are responsible for eradicating viruses, fungi, or bacteria.